what does the plasma, I mean to say, what is the major function of the cholesterol? Very simple point. All the students remember my words. The major function of the cholesterol is fluidity of the plasma membrane. What, friends? Fluidity. You know, some students will think that plasma membrane will attach, will be present very tightly. That is wrong statement, friends. You know, right? Plasma membrane is always moving like this. Always it moves like this. Keep on moving inside the cell. So how it is moving inside the cell continuously? With the help of sterols only. That is nothing but cholesterol. So now every student understand what is fluid mosaic model. It contain lipid, it contain protein, it contain carbohydrate, it contain cholesterol. Okay. Now from this particular slide, I want every one of you pay utmost attention. Okay. So in the BSc and MSc level, you just studied what type of lipids present, what type of proteins present, what type of carbohydrates present. But now we have to study very depth. Now listen everyone. The major chemical composition of the plasma membrane: lipids, proteins, carbohydrates. So friends, I'm telling you, right, when you talk generally means in 80% of the cells, lipids only highest concentration, followed by the protein, then very low concentration will be carbohydrates, and very low concentration will be cholesterol. Not to get confused with that. Now, let's talk about one by one. Okay. Now, I am going to ask you one very simple question every student has to answer. So, plasma membrane contain lipid, protein, carbohydrate, then followed by the cholesterol. Friends, you all people know very well, plasma membrane can perform many functions. Plasma membrane involved in the transport, we will discuss in the upcoming slide. Plasma membrane required for signaling, many functions it will perform. My question to every student is, among these four compounds, which compound or which molecule will perform majority functions of the plasma membrane? My question to all of you is very simple question. Among all these, which can perform majority of functions performed by the plasma membrane? All the students all the time remember, friends, proteins. So please do not get confused that lipids present in the more percentage. Not to confuse, friends. I will tell you in the upcoming slide. Lipids present in the more percentage doesn't mean that lipids are performing more functions. Proteins perform more functions. We will discuss one by one. So all the students has to listen from this particular slide. Now it become little difficult. Follow me correctly. What are the type of membrane lipids? What every student will read in the BSc and MSc level, lipids means abundant amount of the lipids present on the plasma membrane will be the phospholipids. This is what every student try to remember. But friends, listen my statements. Plasma membrane contain, someone is making sound. Please mute yourself. Please mute yourself. Do not make any mistake. Do not make any noise in the middle of the class. It disturbs me a lot. Okay. Look into this. You are all completed MSCs and you don't know how to turn off your audio and video also. So look into this. What kind of membrane lipids that are present on the plasma membrane? So first point every student has to remember, there are many type of lipids are present on the plasma membrane, but every one of you remember, every single lipid present on the plasma membrane, we called it as amphipathic lipid. Now you have to understand why you are calling every lipid on the plasma membrane as the amphipathic. You only look into the picture, friends. What does the amphipathic lipid contains? Every student follow me. It contains a polar head. You can also call it as a hydrophilic head. And what else it contains? A hydrophobic tail. In the lipid chemistry, in the chapter of biochemistry, we read it very correctly. Hydrophobic tail contains what fatty acid? They contain saturated fatty acid. They contain unsaturated fatty acid. Anyway, try to understand me. So it contain hydrophobic tail also. It contain hydrophilic head also. So friends, both the polar part is present and non-polar part is also present. That is the reason only, friends. Every single lipid that is present on the plasma membrane, we called it as amphipathic lipid, not to get confused with that particular point. Right. So listen, what are the type of membrane lipids present on the plasma membrane? See, friends, these three are the major type of lipids: phosphoglycerides, sphingolipids, cholesterol. I will explain you all the structures briefly and elaboratively. I want you to understand, because this information you can't get in any other books except a big reference books such as the Ladish and Carp. Except this, you can't get anywhere. So I want you to understand the information present in the PPT correctly. Phosphoglyceride, sphingolipid, cholesterol. Friends, now let's talk about each and everything elaboratively. First one, phosphoglyceride. You only tell me, how can I define the word phosphoglyceride? It contains phosphate group, and friends, along with the phosphate group, it is having glycerol as their backbone. What it is having, friends? It is having glycerol as their backbone. That's why you can call phosphoglyceride. Let me explain to all the students. Focus. Phosphoglycerides. Why you called it as? Most of the lipid contain phosphate group. Correct, na? It contain phosphate group. That's why you can call phospholipid. Okay, that is good. But then why you call phosphoglyceride? The answer is that all the phospholipids deposited on the backbone are built on the backbone called glycerol. That's why we can call the phosphoglycerides. Every student, please look into the diagram very correctly, friends. Everyone follow the diagram. This is called hydrophilic head. Now you only look into the head. What the head contains? Head contain phosphate group. Correct, na? So head contain phosphate group. That's why we call it as the phospholipid. Okay, you got it. And why you call phosphoglyceride? Every student remember, it contains glycerol as their backbone. So phosphate group is present. Glycerol is also present. That is the reason only we can call it phosphoglyceride. Not to get confused with that particular point. Easy to remember. Okay. So every student understand very easily about phosphoglyceride. And I am requesting every one of you remember the second classification. Second classification is the most important classification. If you get a question from the composition, there is a chance you can expect the question from sphingolipid. All the students try to understand. I will explain in a simple manner. Follow me. What is sphingolipid? I will explain twice. Just follow me correctly. Sphingolipid. How can I define? Sphingolipids are the type of lipids. They are having the backbone. What is the backbone here? The backbone is called as sphingosine. What is the backbone, friends? The backbone is called sphingosine. Now you have to ask me, sir, what is sphingosine? It is not at all required to remember the structure, but it is very important to understand the structure. Every student listen, I will define it in a simple manner. Sphingosine means, every student follow me. It is a long hydrocarbon tail. Long hydrocarbon tail with a polar domain and that contain an amino group. Such a simple definition, friends. Look into the sphingosine definition. It contains long hydrocarbon tail, carbon and hydrogen, which contain a polar domain. That polar domain associated with which group? Amino group. So friends, what is my statement to everyone? Every sphingolipid present in the body, every sphingolipid, it contains sphingosine compulsory. It contains sphingosine as their backbone. Now, every student remember, same structure I am showing you. This is only sphingosine containing a long hydrocarbon chain, a polar domain and amino group. Now every student, please try to understand me. This amino group will form an amide bond. Friends, it is very important for the exam. This amino group will form an amide bond with a carboxyl group of fatty acid. That is called as what, friends? Ceramide. Right. So if you ask me, sir, please give the definition of the ceramide. How can I give the definition? Sphingosine will form an amide bond with the fatty acid of the carboxyl group. That is called what, friends? Ceramide. Don't get confused at all. You have to remember this compulsory. So now I am going to explain to all the students what are the examples of sphingolipids. Remember, what are the examples of sphingolipids? One is sphingomyelin. There are many examples are there. Cerebroside and ganglioside. I will explain all the structures. Don't worry at all. Now you only listen. I am explaining to once again. You know, right? Inside our brain, what we are having? We are having the neurons. You know, right? Neuron contain lipids, right? What do we call the neuron sheath? We called it as sphingomyelin sheath. You know, right? Whichever contain the myelin sheath, we can call myelinated neurons. Whichever do not contain myelin sheath, we called it as what neurons? Unmyelinated neurons. Correct? Or non-myelinated neurons. What is the doubt? Please tell me. If you have any doubt, you please ask me immediately. Okay. Now all the students try to understand. Now I will define you what is sphingomyelin. All the students follow me. Friends, this is what every student. It is a sphingosine. What it is having? Long hydrocarbon tail, a polar domain with an amino group. That is called what, friends? Sphingosine. And when the sphingosine added by the fatty acid carboxyl group, what do we call, friends? Ceramide. Yes or no? Yes. Now, friends, how can I define the word sphingomyelin now? Ceramide, when it is combined with the phosphocholine as their head. What, friends? In a simple definition, ceramide plus phosphocholine. It is called as what, friends? It is called as a sphingomyelin sheath. Never ever get confused with that particular point. Always remember it easy. Now, many times which is asked in the exam is these two examples. One is called cerebroside. Another one is called ganglioside. So what is the difference between the cerebroside and ganglioside? I will give you very simple definition. Cerebroside means, friends, ceramide plus simple sugars. What do you mean by the word simple sugar? You can take a monosaccharide, you can take a disaccharide. Ceramide plus a simple sugar, you can call it cerebroside. Then, friends, what is ganglioside? Ceramide plus oligosaccharides or the polysaccharide such as sialic acid. That is what do we call, friends? Ganglioside. What is the question that is given in the exam? What is the question that given in the exam? Which of the following are the examples of glycosphingolipids? This is the question asked in the exam. Which of the following are the examples of glycosphingolipid? You tell me, friends, why I am using the word glycosphingolipids? You all people know very well, because the sphingolipid associated with the sugar. That's why we can call glycosphingolipids. Every student remember my words compulsory. Glycosphingolipids are abundantly found in the nervous system. This is also direct question you can expect in the exam. Which of the following lipids are abundant in the nervous system? Which is glycosphingolipids. Not to get confused. So, friends, I am summarizing everything in this particular slide. Easy to understand, easy to understand. So this is sphingosine. What is sphingo? Please, no need to focus on the structures you never ever get in your exam. But you have to understand how they look like, that's all. What is the sphingosine? Sphingosine contain long carbohydrate chain with a polar domain amino group. That is sphingosine. When you add an amide bond, it becomes ceramide. To the ceramide, when you join the phosphocholine, it becomes sphingomyelin. To the ceramide, when you add the simple sugar, it becomes cerebroside. You only look into the picture, friends. Ceramide plus galactose, which is the disaccharide, very simple sugar, it becomes cerebroside. Now you only look into this ceramide plus complex sugar like sialic acid. Then what do we call it as? We call it as the ganglioside. Not to get confused with that particular point. I want you to remember this, friends. It's very important. You have to remember it. Okay. So I told you, right, the nervous system is exclusively rich in the glycolipid. Name of that glycolipid is galactocerebroside. Just remember that name enough. Okay, that is fine. So we have studied the first composition of the plasma membrane. That is what, friends? Lipids. Because lipids only present in the highest portion. Before studying the proteins, let's study the cholesterol. Every student has to remember for the exam what kind of question they can give us in the exam. Which of the following cells do not contain cholesterol in the plasma membrane? That is a very good question. Which of the following cells do not contain cholesterol in the plasma membrane? Every student remember, cholesterol is abundantly found in the animal cell. Friends, 90% of the cases, cholesterol is absent in the plant cell also. Cholesterol is absent in the bacterial cell also. Now you can ask me a question. Sir, if cholesterol is absent, then how does the plant cell and bacterial cell will show the fluidity? They are having a different type of lipids. We will explain in the upcoming slides. Okay. Now the question to every student, how cholesterol is embedded in the plasma membrane? This is very interesting statement. You only look into the picture, very interesting it is. Friends, this is the structure of a cholesterol. You know, right? I have explained it to all of you in the chapter of biochemistry also. How does 99% of the lipid look like? Every lipid look like this, which contain polar head, which contain non-polar tail. This is the structure of a lipid. But you see here, how does the cholesterol is looking like? Cholesterol is not like this. Every student has to remember, cholesterol is a ringed structure. You are all able to see, right, friends? This is the ring of the cholesterol. You know, what do you call this particular ring? I want you to compulsory remember. It is called cyclopentanoparahydrophenanthrene. Cyclopentanoparahydrophenanthrene is the name of the ring of the cholesterol. Now you have to ask me, sir, how the cholesterol is situated in the plasma membrane? Every student, please look into the diagram very correctly, friends. This is what group of the cholesterol? Every student, this is the OH group of the cholesterol. Look into this, look into this. The OH group of the cholesterol associated with the polar head of the plasma membrane. Friends, you only look into this. This is what, friends? The tail of the cholesterol. You only look into the picture. The tail of the cholesterol associated with the tail of the plasma membrane. I hope you are following my words. Head of the cholesterol associated with the head of the plasma membrane. Tail of the cholesterol associated with the tail of the plasma membrane. Tail of the lipid. So that is how, friends, cholesterol can easily embedded within the plasma membrane. Friends, most important point, you know, right? All the sex hormones. You take an example, testosterone. You take an example, estrogen. You take an example of progesterone. You take an example of cortisol. You take an example of corticosterol. Friends, these all can easily cross the plasma membrane. Why? Why all these hormones can easily cross the membrane? Because friends, all these hormones, they are having cholesterol as their backbone. Since they are all having cholesterol as their backbone, they can easily cross the plasma membrane. Never ever get confused with that point. See friends, see here, OH group is associated with the head of the lipid, tail of the cholesterol associated with the tail of the lipid. Easy to understand. Okay. And the most important for the exam, where you can expect the question is from proteins. This is more important. So we studied that plasma membrane contain lipids, plasma membrane contain cholesterol. Now what we are left with? Plasma membrane contain proteins, plasma membrane contain carbohydrates. Now the question is very clear. When compared to the carbohydrates, proteins will be present at a high percentage. At the same time, maximum functions are also performed by the proteins only. Not to get confused with that. Now we ought to understand what kind of proteins are present, what are their functions. Once again, I am requesting all of you to remember this compulsory. This is very important for the exam. See, friends, I am telling you one line. I told you this line many number of times. Inside the human body, how many number of cells are present? 37.2 trillion cells are present. Trillions of cells are present. 37.2. So what is my interesting statement? 37.2 trillion cells present in the body. If any cell wants to work, they require protein. So what my conclusion to all of you? Without protein, none of the cell never ever show any function. That's the most important point I'm trying to say. And on the plasma membrane, there is only one protein is present, friends. No, that is wrong statement. What is the statement I have written here? I have written a statement that it contains collage. Collage means what, friends? A group. You can also call variety. Variety of a different proteins are present. Now, I also told to all of you, how can I classify it? Every student remember, based on the function performed by them, first point. Then friends, the second one is based on the structure. Based on the structure, based on the function, there are total three type of proteins are present in the plasma membrane. I will explain you briefly. First one, we can called peripheral proteins. The second one, we can called integral protein. I will explain you now. The third one is called as transmembrane proteins. These three proteins are very, very, very common in the plasma membrane. So friends, now you listen. What functions performed by the plasma proteins? What not, friends? I can say that all function. Every single function is protein only. Every single function. Now, friends, you only listen. How many number of times I have explained it to all of you. If anything want to enter inside, what do we require? On the plasma membrane, what we are having? Once again, in the plant physiology, I'll explain to all of you. And you all people know it. Zoology people read in the nervous system. On the neuron membrane, on every plasma membrane, forgot about neuron, on every plasma membrane, we are having channels. We are having carriers. We are having pump. Friends, what is the function of this channel, carrier, pump? What is their function? They are involved in the transport of an ion. They are involved in the transport of a molecule, either by passive transport or by active transport. That is secondary thing. What these transporters are, my friends, all these transporters are chemically proteins only, right? That is what I am trying to say. All the transporters present on the plasma membrane is protein only. Everything, channel, carrier, pump, all are. Now, we started reading from the 11th standard, all the enzymes are proteinaceous in nature. So, of course, a true statement it is. Every single enzyme present on the plasma membrane, which is absolutely protein in nature. Now, friends, the third one, most interesting one, receptor. How many number of times we discussed inside our body, anything want to show the function, any ligand. Friends, let's take an example, hormone. Let's take an example, a growth factor, right? Let's take an example, neurotransmitter. Friends, anything, if anything wants to work in our body, suppose, friends, this is a hormone. Example, let's take insulin hormone. Otherwise, let's take a ethylene hormone. So, I think I am audible to everyone. When you just refresh yourself. Refresh. I think it's problem from your side. Okay. Now listen all of you. So friends, if any growth factor or any hormone want to show their action, it has to associated with what? It has to associated with the receptor compulsory. So friends, what is receptor? That is the question here. Friends, receptor nothing but what? Protein only. A special protein present on the plasma membrane, which is providing the binding site to the signal molecule. The signal molecule may be growth factor, signal molecule may be hormone, signal molecule may be neurotransmitter, doesn't matter. Whatever the signal molecule, if it want to show the function, it has to associate it with the receptor. So friends, what is that receptor? Which is the protein. Now you only listen, my friends. If one cell want to be connected with the another cell, we can call cell-cell recognition. There also proteins are required. Very important point. And not only that, the very interesting point. If the cell want to connected with the extracellular matrix. Every student know, right? You all studied in the MSc level. What is extracellular matrix means? Which contain collagen, which contain nidogen, which contain keratin, which contain elastin. These are all what proteins? Extracellular matrix proteins. Friends, listen. This is one cell. This is another cell. Here cell joining. But here cell is joining with matrix. See, not to confuse friends. Every student follow me. This is the cell. This is the cytoskeleton. And this is what? Matrix. You know, right? What does the cytoskeleton consist of? It consists of actin filaments. It consists of tubulin filaments. So friends, cell is connecting with the cytoskeleton also. Cell is connecting with matrix also. I will also give you the definition. Cell is connecting with the intracellular environment. Cell is connecting with the extracellular environment also. How, friends? With the help of proteins only. So what I am trying to explain to all of you, what not? Every single function inside the living cell is mediated by the protein only. So similarly, on the plasma membrane also, every single function is under the control of protein only. Never get confused with that. See, in this diagram also you can see here. This is transporter. This is also transporter. We can call it them as channels, carrier, pump. They are all what? Proteins. Now you see, friends, this is the protein. This is what? Cytoskeleton. And this is what? ECM protein. You only listen, understand. So the protein connected to the extracellular environment, protein connected to the intracellular environment also. And protein can act like a receptor, protein can act like a enzymes. Every function is under the control of protein only. And friends, all these proteins which are present on the plasma membrane, they are broadly categorized into how many types? 3 types. Every student remember, how did we categorize it into 3 types? Based on the structure, based on their arrangement on the plasma membrane. Third and most important one is function. So friends, based on the function performed by it, based on their arrangement, based on their structure. What are the three types? Peripheral, integral, transmembrane. 90% of the students will get very much confusion between integral protein and a transmembrane protein. Now I will give you the differences and now you have to tell me the statements are true or false. Okay, first let's talk about peripheral proteins. Friends, what did I told you about the peripheral proteins? Suppose this is the lipid bilayer. Okay, this is the plasma membrane. This is what, friends? Lipid bilayer. Very important and interesting statement. So where does the peripheral protein attaches here? Here, peripheral proteins are present. Now every student listen. The question for the exam point of view is, which of the following proteins can be removed easily from the plasma membrane? That is the question. Which of the following proteins can be easily removed from the plasma membrane? Undoubtedly peripheral proteins. You have to ask me why. What is the reason? The reason is that peripheral proteins just associated with the hydrogen bonding. That's all. So every student commonly know that hydrogen bonding is very easy to break. So friends, you can easily separate the peripheral proteins. No need to get confused with that. But you only look into the next one, integral membrane proteins. All the students has to understand the picture very correctly here without confusion. See friends, this is the plasma membrane. Integral protein embedded within the plasma membrane, You know, right? Every student know this. They are called alpha helix. You don't need to read that, but just I'm telling you for your understanding. So what I'm trying to say, the protein is embedded within the plasma membrane, very deeper. So friends, you can't separate this very easily. So what you have to use, that is the question. If you want to separate this integral proteins, we should always use the strong detergents. You have to use the strong acids. Then only, friends, you can separate. If you are doing a PhD, in the PhD, you will do all these experiments. Separation of the proteins from the plasma membrane. You can always use the strong detergents or the strong acids. You can separate it easily. Now, all the students remember my words correctly. What is the difference between integral protein and a transmembrane protein? See, friends, listen. It's a very simple definition. This is a protein. Where it is present? It is present or embedded. Embedded means, friends, incorporated within the plasma membrane. So it is incorporated within. So it is called integral. No doubt about it. No doubt. So friends, you only try to understand. Very good. If this particular same integral membrane, if it can move on the membrane, so like this, if it is moving on the membrane, then what do we call it as? We call it as transmembrane protein. So friends, now what is the definition I am going to give you? Remember, very important statement. All transmembrane proteins are integral proteins. This is the very important statement for exam. But all integral proteins are not transmembrane proteins. Friends, what do you mean by that? You only tell me this is a integral protein. Just understand. It is not moving on the plasma membrane. It is not moving like this. Then what I can say that? I can say that is only integral membrane. It may move on the plasma membrane or may not move on the plasma membrane. Suppose, friends, this is the plasma membrane. It is the transmembrane protein. My friends, the first point is what? If the transmembrane protein want to move on the plasma membrane, first of all, it has to embedded inside the plasma membrane. So friends, every transmembrane protein is an integral protein. But every integral protein is not transmembrane protein. That is important for the exam. Why, friends? This is integral protein. It may move or may not move. But If a transmembrane protein want to be moved, first of all, it has to embedded inside compulsory. So that's why what we can say, all transmembrane proteins are integral membrane proteins, but not all integral membrane proteins are transmembrane proteins. This kind of statement is common. Please mute your mic. Don't make noise. Okay, okay. Is it clear, everyone, so far? Just let me know. Is it clear, everyone? So please mute your mics. Mute your mics. If anyone having any doubt, I will re-explain. Okay, that's all for now. We will meet in the tomorrow class. So botany people having 7:30 madam's class, I will explain the procedure.